depending on wherever you are. Welcome to the ASLM special COVID-19 session number 4, an initiative of the African Society for Laboratory Medicine, Africa CDC, FIND, ICFIA, the Korea World Bank Partnership. My name is Anafi Mataka from ASLM, currently serving as a project lead for accelerating access and integration of innovative diagnostics funded by Unitaid, as well as a project manager for the laboratory systems strengthening community of practice funded by the Bill and Melinda Gates. Some logistics. This will be a 1 hour long session, and we have lined up 2 presentations. There will be 15 minutes each, after both of them will be followed by a question and answer segment. Please kindly remember to mute yourself when you're not speaking so that we avoid the background noise. And questions or comments, as usual, in the chat box. Session proceeds. Colleagues, as you know, laboratory testing has been an integral part of the strategy for countries' preparedness, alertness, and response to the identification, management, as well as care for COVID-19 cases. In response, ASLM and FIND are dedicating this session really to focus on COVID-19 diagnostics manufacturers for a couple of few that are going to be coming, for which we know there's still so much knowledge to be acquired. We are so privileged today that key to this fight, manufacturers are also found at the heart of it. And today, we appreciate them taking some time out of their busy schedules. The first set of manufacturers today will be BD, Becton Dickinson, as well as Cepheid. They will specifically talk to us about what they are offering, particularly technical issues regarding their various technologies. We shall look into their overview of the COVID-19 lab tests that they are offering, things like what genes are being targeted for, the status of regulatory approval, the performance and runtimes considerations, as well as information about training and resources. Would like to set the record straight as we discuss that diagnostic manufacturers are also an industry. But today, we are not using this as a marketing platform. And as a result, we will kindly ask all of you that we may not discuss issues of pricing, issues of maybe supplies in a way that will be more competitive. The way we would want to discuss today is really technical issues and what we can leverage on as we fight this deadly pandemic. We shall start with a presentation from BD. And our first presenter from BD is Dr. Charles Cooper, affectionately known as Chuck. At BD, Chuck oversees the clinical development of a broad range of infectious disease diagnostics for tuberculosis, sepsis, sexually transmitted diseases, bacterial vaginosis, as well as cancer screening. He's a board certified in infectious diseases, and he has a specialty in certification, and he has a specialty certification in HIV medicine. In his course of his career, he has served in various leadership roles in both the US Food and Drug Administration, FDA, as well as BD, while continuing to providing care to HIV patients in the Baltimore and Washington area. Chuck's clinical interest is in HIV medicine and is a practicing infectious disease specialist who currently provides clinical care for underserved patients at the federally qualified community health center, Greater Baden Medical Services. I would like to hand over to Chuck to take us through the first presentation.
Thank you, Nafi, for that kind introduction. I feel very honored to have this opportunity. So let me see if I can share my screen. Let's see. Here we are. Are you able to see my slides?
Yes, Jack. You need to put them on
Yes,
presentation.
yes, we can see.
Yes, Jack, we can see the slides. Thank you.
Great. Thank you. Thank you so much for inviting me. This is
Yes, you can see.
okay, great. Excellent. Thank you all. Thank you very much for inviting me to participate. This is a great opportunity, and any opportunity to share knowledge with regard to this pandemic of any knowledge, I think is a valuable opportunity. So Ms. Chuck Cooper, Medical Affairs Department, Integrated Diagnostic Solutions. So I'm not R&D, so the usual highly technical questions that I may have to follow back up on, but I know the basics, and I can describe for you our diagnostic platform as well as some of the clinical considerations. We focusing mainly on the molecular solution that we have, mentioned also some other things that we're pursuing. So for the agenda, we'll start with just a quick overview of the BD mission. We'll talk about the BD MAX system and the specific SARS-CoV-2 reagents that have been developed. And that's where we'll spend most of the time, but also spend some time talking about specimen types, the results, and what the algorithm is, some limitations, other diagnostics that BD has, questions and answers. So BD is very much of a global company, and very much has a history of being dedicated towards increasing access to products and healthcare solutions that improve outcomes, protecting patients and healthcare workers. This has been a long time focus for BD. So how is BD mobilizing for the COVID-19 pandemic? So I think one of the things that's really interesting is that BD has a really broad array of medical devices and technologies, and this epidemic is really touching all of BD segments. There are information systems that BD has that help track the spread of the virus. We have things like IV pumps and of course vaccination items like syringes. Also, of course, we have diagnostics, and we collaborate very actively, Renuka and Gary, with global health agencies. So we're very, very much actively involved on a number of fronts, doing everything we can to help address this pandemic. So let's talk about the BD approach to COVID diagnostics. We'll focus mainly on molecular, but then we have some development activities involving serology and antigen detection that I will mention briefly, just so that you're aware. So the BD MAX system, it's a highly automated system, and with that automation comes standardization, efficiency, and convenience, and that's one of the key sort of values of the BD MAX system. How does it work? Well, it works with the unitized strips, and this is the picture you see here is an example of a unitized reagent strip, and there's a different strip for each diagnostic test that you might want to run. So we have an MDR-TB assay, and that has its own strip, and we have things like carbapenemase resistant testing. We have MRSA, enteric pathogens. These are the sorts of tests and so we've developed one of these for SARS. So what does the cartridge look like? Where does it take place? Well, the reaction takes place in these little credit card-sized cards. They're called, we call them, and you see each of the wells is a 4 microliter well, and there are 24 of them. So this enables you to 24 actions at the same workflow look like. So workflow essentially it's less than 1 minute of hands-on. Essentially add the specimen to the sample buffer tube. You add the tube to the rack. You load this unitized reagent strip, and then you also place this PCR cartridge, and then you're done and essentially push the button. And then it's a simple and fully automated, which makes it very useful. It's a top instrument, so it sits on a, you can sit on a desktop. It doesn't take up a huge amount of space. It also has this feature that makes it fairly unique, which is that it has an open system capability. Research academic institutions, laboratories have the opportunity to their own developed assay that they can then run on the Macs. And so we've seen a lot of interest in that to pandemic with a lot of labs that have assay early on, prior to the availability of commercial assays. A lot of labs were developing, which was, I think, was pretty neat to be able to support. So pre-analytical considerations are critical. Just like with any assay, it doesn't matter how good the assay is or the technology if the specimen wasn't collected well. And so collecting the correct specimen and using the correct device actually makes a big difference. And with BD Max, as with any diagnostic platform. I think this is an interesting slide, and I think, you know, what this slide points out is, with regard to specimen collection, where do you collect the specimen for SARS-CoV-2, for COVID-19? And so there's been a lot of publications and discussion about where you collect the specimen. And, you know, this has to do with ease of collection, simplicity, reducing barriers to collection. Nasopharyngeal collection is more difficult. It's difficult to consider a patient could do a self-collection. It can be challenging, but it turns out that oropharyngeal and nares collections may function just as well. They may perform just as well as a nasopharyngeal. One of the things that's different about this COVID-19 from the original 2003 SARS is that there's quite a bit of viral replication happening in the upper airway. And that's different than the 2003 SARS, which didn't really have quite as much viral replication. And so the other thing is that viral replication is happening very early on in the disease course. So at the time of symptom onset, patients have a very high level of viral load in their upper airway. And so that makes it easier to actually perform these kinds of collections and perform diagnostic testing. But many have speculated that high viral load in the upper airway early on in the course of disease is one of the key reasons why this infection is so transmissible. It makes it easier to transmit. So I think that's an interesting topic to consider. You see the specimen transport approach in the lower right-hand corner, particularly different than their diagnostic tests. So let's turn to the actual reagents. So there are actually 3 different approved tests that have been developed for the BD Max. SIR-test was the first one, and it was CE marked on March 9th. The assay design focuses on the spike protein gene, the S gene. It's a single target assay. It's distributed by BD in Europe, and there's an option for it to be also distributed in other countries outside of Europe that do recognize CE mark. The second assay that was developed and released is by BioGX. So BioGX and SIR-test are small companies that actually developed commercially available tests that are able to run on the Macs. And they're sort of taking advantage of that open system capability to develop a commercial product that could run on the Macs. And that's what SIR-test did. And BioGX developed a SARS-CoV-2 diagnostic and achieved FDA emergency use authorization. The assay design is different. It actually uses a 2-target design that's consistent with the CDC recommendation. That assay is only available for distribution in the US. And the third assay I wanted to talk about is the BD developed assay, which has been approved by FDA for EUA, and it has also received a CE mark. So it's available in the US and outside the US as well. So for that third one, let's talk a little bit more about it. So here's some more information about that test. You could run 1 to 4 samples in 90 minutes. If you're going to run 24 samples, it will take as long as 3 hours, but it can run anywhere from 1 to 24 samples. Moderate high complexity laboratory setting is where it is. It's a qualitative assay, and it accepts specimens from nasal collection, nasopharyngeal, and oropharyngeal. So what about the results? So this slide is a little busy, but I think it's important to understand the design of the BD assay. Assay targets the nucleocapsid N1 region, as well as the nucleocapsid N2 region. But the way the algorithm is set up is designed to be maximally sensitive, so that for algorithm, if either N1 or N2 fires, you get a positive result. And this is intended this way to maximize sensitivity of the assay. I think that's the main point for this slide. Yet, what the percentage of specimens would have just N1 or N2. We're sort of learning very much as we go. The development program is obviously rapidly accelerated and learning as we go. So a positive test for COVID-19 means that RNA is present and the patient is infected. Mean that they're contagious because we think that later on in the course of the disease infection, patients may still be PCR positive but may not quite be infectious. That's a story that's holding. A negative test doesn't necessarily completely rule out that infection is present. This is the clinical trial data that we have. We did not run a prospective clinical trial because that would take a long time. So what we did was a spiked contrived specimen trial where we were sampled near the to really challenge the assay. And you see we did 40 replicates at 1 to 2 times the limit of detection and had a 95% sensitivity. At 3 to 5 times the limit of detection, we had 100% sensitivity. So that gives you an idea of the data that was used to achieve approval. So limitations, there are always limitations, and these are not unique to this particular test. Proper sample collection. I think what is unique is sort of the unfolding story as we learn more about the pathophysiology of this disease. Our ability to interpret more effectively the results of any diagnostic will improve as we learn more. So now turning to just a couple of slides on other development programs that we have, just to share briefly. We are working on immunoassays for the diagnostic testing of COVID-19. So this is the Veritor, and it's a lateral flow immunoassay reading system in this small handheld instrument. We're also working on a fully read lateral flow assay as well. These don't have the sensitivity of a PCR, but could be helpful in an outpatient setting with patients that are less severely ill, and sort of we're hoping would divert demand away from PCR testing and molecular testing, which could be used for patients in the hospital. So we've entered into agreement with Biomedomics, which is a company in North Carolina, to help them with their assay and distribute their assay. And we're working to develop evidence to help expand our understanding of these serology tests. This assay detects IgM and IgG antibodies against SARS-CoV-2. It can use either whole blood from a finger stick or serum or plasma. And serology assays really will expand our understanding of what's happening with COVID-19. We don't understand the spread of this pandemic because it turns out that a lot of patients have mild infections, and some significant portion will even have asymptomatic infection. So the question remains, what percentage of the population of any community has been infected? These serology tests will help us understand that. And so these are being rolled out right now as we speak. Biomedomics is submitting an EUA to FDA, and there are numerous other companies that are also working to make these sorts of serology assays available. We don't know yet if a positive serology correlates with immunity, but that's something that's being explored, and researchers are working hard to understand what the correlates of immunity are. So that's what I had to share today. I guess, are we doing questions now, or will we wait to the end of the second presentation?
We'll wait until the edition.
Okay, thank you very much.
All right, thank you very much, Chuck, for the wonderful presentation. As we've just mentioned, we will hold on to our questions until we finish with the second presentation. Chuck, you can stop sharing. Our second presentation is from Cepheid, and our presenter will be Rujeko Tsomondo. Rujeko is a regional application lead for Cepheid, who supports countries in Africa with training and implementation of the GeneXpert system and tests. She is involved in creating training materials and delivering training online, in country or at Cepheid training centers. Before moving to Cepheid, Rujeko worked as a biomedical scientist for NHS England. She inherently understands the importance of technical training for laboratories as a valuable asset and is driven by this in her daily activities. Rujeko is also passionate about global health. Ladies and gentlemen, allow me to welcome Rujeko to give us a presentation.
Okay. Hi, good afternoon, everybody. I think Chuck, is he still sharing his screen, or can I share my screen now?
Yeah, share your screen.
Okay. It still says like...
Chuck, can you maybe, yeah. Thanks.
Okay.
Thank you. Go ahead, Rujeko.
Okay.
No.
Good. Wonderful. Go ahead.
Okay.
Yes.
All right. Thank you very much, ASLM, for giving us this opportunity to speak about our system and our test. As we prepare laboratories for testing, that my colleagues are online. So if you have any questions while I'm presenting, please feel free to write them in the chat. And my colleagues will also be helping me to respond to your questions. So with this invitation and this presentation, I will be answering the questions. So I will give, first of all, an introduction to the platform, so the GeneXpert platform, and also update you on the other tests that can be performed on this platform. Then we will look at the COVID test and what genes are targeted, really the technical information that you need to know, an update on the regulatory approval, the runtime, technical considerations that you need to have in place, and any requirements or consumables that are not supplied with the tests. Finally, I will speak about the performance of the test and also training and some links to resources that are available to you right now. Okay. So when we talk about the GeneXpert system package, the system itself consists of the modules. So here we see a GeneXpert with 4 modules and a computer system with either, which would be either a desktop, and that has the GeneXpert software preloaded. Along with that, there is also what we call a assay definition file. And this allows the system to recognize the cartridge that is being run. Can everybody hear me?
Yes.
Yeah,
Go
go
ahead,
on.
Ricky.
I can hear you.
We can hear you.
All right, thank you. A barcode scanner, which is required for doing the tests and loading them onto the system. So the GeneXpert system requires the use of single disposable cartridges. And that means that one test will be performed and the cartridge will be disposed. The cartridge is self-contained, meaning that they contain all the reagents that are required to perform the test. There are also accessories that we recommend. So where there are electricity supply shortages, a UPS or surge protector, batteries or generator are recommended. This will ensure the functionality of the system. Optional accessories include a printer, just in case the printing of results is required. So when we speak of the GeneXpert system package, we speak about everything that we see here. So now we see the Cepheid test menu, and on the left, you see the tests that are currently available. All these tests are tests that can be run on the GeneXpert system. You can see that there is also projecting into the future the tests that are coming. Our tests are focused in 5 groups. So healthcare associated infections, critical diseases, which includes our SARS-CoV-2 cartridge, women and sexual health, virology, and oncology. So all these tests that you see here can be performed on the GeneXpert system. So what is the solution? Cartridge, which is the Xpert Xpress SARS-CoV-2. And just to clarify that SARS-CoV-2 is the virus strain that causes the disease COVID-19. So this cartridge here that you see received emergency use authorization from the FDA, and our CE IVD marking is expected in the second half of April. The test detects 2 targets, the E gene, which is an envelope protein, and N2, which is a nucleocapsid gene. Also included inside the cartridge are onboard controls. Because these controls are in the cartridge, that means that each sample
E and N2, it one time.
Cartridge.
E and N2 would means E and N target.
Please, can everyone who is not talking mute their?
Yes, if you're not, please mute your microphone. Thank you. So there are controls included inside the cartridge for each sample, meaning that each sample that you process will have controls to check for that reaction. There is the probe check control, which verifies the reagents, the probe integrity, and also the dye stability. And then there is the sample processing control, or SPC, which verifies the adequate processing of the sample. So it monitors for inhibitors and it ensures that the real-time PCR conditions are appropriate for the reaction. The system is a closed cartridge system, and this helps to minimize the risk of contamination, meaning that once the sample has been added to the cartridge and the lid is closed, the sample will not have any contact with the inside of the instrument. Looking at the run time, the test completes in approximately 45 minutes. And we currently have early assay termination, which is being submitted for regulatory approval, meaning that result would be reported sooner than 45 minutes. But that has not yet received regulatory approval. And once that is done, we will update you. There is access to on-demand results, meaning that at any time when the tests are running, you have access to any results that you would like to look at on the system. And random access, meaning that each module on the system, on the GeneXpert system, is independent of the next. Run different tests on the same system. So this allows, for example, a site that normally runs the TB cartridges to also use any of the modules to run the SARS-CoV-2 cartridges. So in terms of preparation for the laboratory, the GeneXpert software has to be on version 4.7b or higher. If it is a site that has the GeneXpert Infinity, this has to be on Expertise software version 6.4b or higher. Just a reminder that these cartridges have received emergency use authorization only, and that we're still waiting for the CE-IVD registration, which will be later this month. For the materials that are required but not provided, allowing for the lab to prepare, would be the sample, and I will look at these in the next few slides. But also, the sample needs to be collected in 3 mL of viral transport media. Of course, there is personal protective equipment to consider, and we recommend a 1 in 10 dilution of bleach and 70% ethanol or denatured alcohol to clean up any spill. I already spoke about the power supply and power backup and the printer in the previous slide. In terms of what the kit looks like and the kit contents, so you see here a picture of the kit contents. There will be 10 cartridges in the box. Also included is the kit CD, which contains the assay definition file and the instructions to import the ADF into the GeneXpert software. You can also see that there is a pipette which is provided, and this pipette will allow you to transfer the sample, the patient sample, into the cartridge. Those pipettes are disposable, and there will be 12 of them in each box. So there are 2 extra pipettes in each box. The storage temperature for the kit is anywhere between 2 and 28 degrees Celsius. So now looking at the specimen types, the assay has been or can be used with 5 different specimen types. So these can be a nasopharyngeal swab, a nasal swab, mid-turbinate swab, or a nasal wash or nasal aspirate. Once these samples have been collected with the swab, they need to be transferred into the 3 ml viral transport tube, which is going to help to preserve the specimen for transportation. Alternatively, if the sample that has been collected is a nasal wash or a nasal aspirate, 600 microliters of that sample should be transferred to the viral transport medium tube, and then the sample will be ready for testing. In terms of guidance for training and for collection of the samples, the images that you see here are included within our training materials on how to collect the different sample types. So here we see a nasopharyngeal swab collection procedure. a nasal swab procedure. And for this one, the nasal swab collection is from both nostrils. Then we see a mid-turbinate swab collection, and that's the last one. Our training materials also include the cartridge preparation steps, which are very simple. So once the sample has been collected, it will need to be mixed. So invert the tube 5 times. And once you're ready to prepare the cartridge, then open the cartridge lid, and using the pipette that is provided in the kit, transfer 300 microliters into the cartridge. Then close the cartridge lid and start that test on the GeneXpert system. So in terms of handling of the sample and loading it into the cartridge, these are the steps that need to be performed. So what will the results look like? Just trying to... So here you see an example of a positive result. In this case, a positive result will be reported where either the N2 or both the N2 and the E targets are detected. The example that you see here, both the E and the N2 targets are positive. However, even if a sample has only the N2 target positive, it will be reported as a positive result. So here we can see the final test result and the PCR graphs. And that's for an example for a positive result. For a negative result, it means that both the targets, so neither E nor N2, are detected. For this type of a result where our target is not detected, our cartridge contains the SPC control, which I mentioned earlier, which validates our negative result by checking and confirming that all the adequate processes in the cartridge went well. So if there is a negative result, the SPC, our control, has to pass, and that will allow for a negative result to be reported. The third example is a SARS-CoV-2 presumptive positive result. Now, a presumptive positive will indicate that only the E target has been detected, like what we see here. Our E target is positive, but the N2 is negative. The reason for this is because the primers and probes for this E target are not specific to this current SARS-CoV-2 virus only, and therefore the result would be reported as a presumptive positive and will require either retesting or confirmatory testing. So the E primers and probes will detect SARS-CoV-1 and other viruses, SARS-CoV-2, which infect humans. That's why the result would be a presumptive positive compared to a true positive that we saw earlier on. So this will actually require the laboratory to retest or to use any other confirmatory test to determine the final result for the patient. So looking at the analytical sensitivity, this was done with SARS-CoV-2 reference material, for which the limit of detection for the assay is 250 copies per mil, but also with live SARS-CoV-2 virus, and the limit of detection here was 0.01 PFU per mil. This information here can be found in more detail in our package insert, and this is on our website, and I will show the link right at the end. For analytical specificity, 7 microorganisms from the same genetic family, and 32 high priority organisms were tested. And from that, there were no potential unintended cross-reactivity. Also, to note is that there is more information now in our package insert, which is available online, if you want to see a list of the different organisms that were tested.
I want conference, but let me.
In terms of biosafety and disposal, the used cartridges and unused reagents need to be disposed of as per WHO recommendations if there aren't any national or regional guidelines. So they should be treated or disposed of per WHO medical waste handling and disposal guidelines. And currently there are updated guidelines on the WHO website relating to PPE and to laboratory biosafety. For commercially available controls, SeraCare controls can be used as the external control for this test. We are currently investigating other sources of external controls, and these will be updated in our package insert once that detail has come through. If you do have questions, though, regarding to these control materials, then the Cepheid team, technical support team, or the medical scientific affairs team would be able to help you with more information about those controls. In terms of training and other online resources, we have prepared training materials in the form of videos, cartridge preparation cards, guidance for sample collection, and also a technical training presentation. And these will be shared for you to use. In addition, we currently have a website which is updated regularly with all the relevant documentation that you need. For example, there is an FAQ section. You have the latest package insert, fact sheets, and safety data sheets available on this online resource on the Cepheid website. And with that, I hope this information will help you to prepare your laboratory for testing, and I'll hand over back to Anafi.
Thank you, Rujeko, for the presentation. We are slightly behind time, but we have about 20 minutes for questions and answers. Quite a lot of questions indeed in the chat box. So my colleagues here will be helping me randomly pick questions as they come. So I'll start with a question from Jibrin Kama. What is the appropriate storage for collected samples as they are transported to testing labs? And I think this will be applying to both the BD and the Cepheid. All right. I'll take maybe 2 questions at a time. So the first one was on specimens. Then the other one is directly to Dr. Cooper from Prof. Tahir Pillay from University of Pretoria. And Prof says, does the open system architecture of the BD instrument allow one to use homemade reagents for COVID-2 detection given the current issues with shipping and shortages of reagent kits? I'll repeat that again. Does the open system architecture of the BDC instrument allow one to use a homemade reagent for COVID-2 detection given the current issues with shipping and shortages in reagent kits? Chuck.
Yeah, yes, it does. BD has those reagent strips that are intended specifically to support development of the sort of a homemade test, so that it is available, and some laboratories are actually have developed their own test using that system.
Thank you. In mind with the issue of the specimen collection asked, I would also want to shift to Rujeko and ask for Cepheid if the Xpress SARS-CoV-2 yields a result negative for N2 target, but a positive for the E target, which is considered result positive, and how many times can the test be repeated for confirmatory tests?
Okay, so I need to respond first of all to the specimen collection, transport, and storage. So the samples I spoke about, which was the nasopharyngeal, nasal, and mid-turbinate swabs, nasal wash, nasal, can be stored at room temperature, meaning 15 to 30 degrees Celsius for up to 8 hours, or refrigerated, meaning 2 to 8 degrees Celsius for up to 7 days until testing is performed.
Thank you.
And then the other, coming back to your question.
Yeah, for the genes.
Okay, so for a presumptive positive, it means that only the E target has been positive, and that will be our presumptive positive. For our positive result call out, we can have either the N2 target positive or the N2 and E positive. The E is not specific to SARS-CoV-2, and that's why that one will not be reported as positive, but a presumptive positive. In terms of retesting, we advise to retest at least once, and then any other confirmatory tests can be used.
Thank you, Rujeko. And I think the next one is from Sheila Chibore. And I think it was directed to Chuck. Do you require different software versions for results analysis for each test? And I would also ask you to respond to a second question from the same person, asking about the throughput of the system per 8-hour working schedule, keeping in mind we need to ramp up testing in all community settings. Chuck.
So there is an assay definition file that is loaded when on the Macs. So there is a update that occurs when someone wants to start testing using this assay, and so it's fairly simple and happens one time at the beginning. For how many specimens can be run, it's roughly about up to 120 per 8-hour shift. And then I think the other question about the storage, 2 to 8 degrees, we're essentially following the standard recommendations that organizations like CDC have issued, which involves 2 to 8 degrees for 72 hours and minus 70 if longer, if it's going to be stored.
Please go ahead, Chuck. Colleagues, if you are not speaking, kindly mute yourself.
So was there another question regarding the throughput? I think there was a question on throughput, which I answered as 120 per 8-hour shift.
Yeah, okay. Thank you for that. And the next question, I think is applicable to both. What are the most common mistakes that health workers are making when collecting samples, whether nasopharyngeal or oral nasal samples? Any recommendations? What are BD or Cepheid thoughts on self-collection by patients? Rujeko?
So far, that has not been verified by Cepheid yet. So the sample types that I spoke about earlier are the sample types that have been verified. So I can't really say that I would recommend that. But if there is questions regarding other sample types that might be considered suitable within the different environments, we have a medical scientific affairs team who can help you to and advise you on the different sample types that you might have in mind. So my colleagues will share that email in the chat.
Thank you. I'd like to take this question. Wilbur has been trying to raise his hand some time back, and they had asked him to jot it down. The first question, or this one, like we said, we are not going to talk around issues of cost for now. But then he says, can speakers address the question of sensitivity for PCR test, given the concerns that are coming through of many false negative tests in social media?
So I think one of the challenges is that sensitivity and specificity are performance characteristics that are usually determined through a prospective clinical trial. And because of the urgent need and the way these assays were developed, prospective clinical trials were not conducted because that would take really too long. So, you know, exactly what the sensitivity and specificity is for a variety of conditions and different sample types is a story that's still unfolding. And so in place of a large prospective clinical trial, we have these spiking studies where we spike and challenge the assay near the LOD. And that's what we have right now in place of sensitivity and specificity.
Thank you. And the next question is really a summary of quite a number of questions that are coming, where people are asking whether sputum is a suitable sample.
Well, I can tell you that our assay was not validated on sputa. We did not actually use sputa as one of the samples that was used during our spike studies. So, you know, I can't comment on our assay and sputa. I do know sputa has just a couple general comments. Several publications have shown that there is quite a bit of high viral loads in sputa. I think one of the concerns about attempting to collect sputa that I've heard from clinicians on the ground is the potential increased risk for transmission or spreading the virus in some cases. But I can't really comment much more than that.
Okay, thank you.
Yeah,
thank you.
for my part, for this.
Anafi? Hello? Anafi?
Martin, would you allow Rujeko to respond on this?
Yeah, just about this.
Okay, so in terms of sensitivity, just like what Chuck was describing, what we have is data based on pooled nasopharyngeal swab clinical matrix. And for Cepheid, that has been done with SARS-CoV-2 reference material and also live SARS-CoV-2 virus. And for more details about that, it's also detailed in the package insert exactly how it was done.
Okay, thank you. I'll take another question for both BD and Cepheid. Does the SARS-CoV-2 PCR test require Biosafety level 2 lab to conduct the test, or can it be done? Okay, this one says SME Microscopy Center.
I think that's a question that's come up, you know, quite a bit. I think what most laboratories, you know, that we're aware of, or a lot of laboratories are doing is for the BD Max, it's really the point of potential risk could be when the specimen is transferred to the sample buffer tube. And so what a lot of labs are doing is doing that transfer step from the specimen that was collected into the sample buffer tube in a hood, in a BSL-2 type hood. And then once it's in that sample buffer tube, then it's loaded on the machine, and then it goes from there. But, with regard to specific recommendations for different settings, we would have to work on that and to provide more information.
Thank you very much. This one has to do with I think test algorithms. So it's from Sunita and says, in the test algorithm, when should the serological test be introduced? How long does the IgM antibodies? I think we have been discussing this one for quite a while. I would like to pass the bit because I think we have another session dedicated to around serology. Another question from Chaba from Chai, Nigeria. Do samples need to be batched? If just 3 samples are run in 90 minutes, are the reagents wasted? How many controls are in each batch?
So,
I guess this will be applying to the chart.
So that unitized reagent strip, each one is an individual specimen. So you only load the number of reagent strips based on the number of specimens you're going to run. So if you run 3 specimens at the same time, you load 3 reagent strips. And so you can go up to 24. The specimen processing control, it's present in the extraction tube, and it undergoes extraction and amplification, at the same time as the assay. So as the assay is run to monitor for things like inhibitory substances and any instrument or reagent issues. So.
Okay. Rujeko, there was a question on biosafety cabinets. Maybe you could respond to that.
Yes, so our advice is to consider the WHO recommendations for processing for nucleic acid testing for COVID-19. And this is a BSL-2 cabinet recommendation or a shield. So we recommend looking at the WHO guidelines, which are being updated regularly. So if that changes, then it would change according to that.
Okay. All right. As I think time is not on our side, I would really request that Rujeko and Chuck, if you can help us maybe as soon as possible. We're going to forward all these questions. I think most of them have not been responded to. If we could go back to them, and then we send them back to the people who attended this session, as time is really not on our side. I would like to take, I think, the last few minutes just for a few words of closing or remarks from any of your colleagues. I know Chuck is with Renuka and Rujeko with Charles. Let me just give either of your teams any 1 minute each for any remarks. Chuck, Renuka.
So maybe I'll just add my appreciation to this wonderful session. Thanks ASLM for taking the leadership in pulling together an industry response with, I've seen that there are at least about 800 people on this call, which is an amazing outreach. We really appreciate it. We've taken notes of the many questions that have been posed, and we will send in our typed responses to you. And you should feel free to circulate this going forward to the audience. This was very helpful, useful way to introduce what's going on in various parts of the world and technology. So thank you.
Thank you, Renuka, Rujeko, and Charles.
Thanks very much, Anafi. Just a quick note, just to say, reiterate the same things that have been emphasized by Renuka. This is amazing, and this is good partnership. And we look forward to working with all the country programs represented here, going into the future and fighting this terrible pandemic. Thank you.
Thank you, Charles. We'll try our utmost best to get back to you with the rest of the responses as soon as possible. And I would also like to announce that these are the first two of similar sessions. On Thursday, we have Thermo Fisher and Kayagen. So please make a date. We are going to be sending the invitations immediately after this call. And we hope to see you in large numbers as usual. Ladies and gentlemen, again, it's the top of the hour, and we know you are busy. You have quite a lot to do, but we will continue to work together until we win this fight against COVID-19. And with these words, I'd like to say goodbye, good afternoon, good morning, and good evening.